again. Welcome to Phage Fridays. This is a room that we do recurrently. Sabrina's on Paul. Every Friday under the Phage Club and Clubhouse, and we get to discuss a lot of topics that are of interest in the Phage community. And today we get to talk about something that's uber cool for me, and I think for a lot of people, which is Phage training and how to train your Phage. That sounds like How to Train Your Dragon, which is absolutely awesome. Up here joining us today is, well, I should let people introduce themselves because I suck at introducing people, but most of you guys already know me. I'm Adriana. Sabrina, do you want to introduce yourself?
Sure. I'm the Research Development Director at Taylor. We're a service center that provides bacteriophages for compassionate use care.
And Paul.
Hi, I'm Jean-Paul Birnet. I'm a Phage researcher at the PNAS Military Hospital in Brussels, Belgium.
We're also joined by two other people from the Taylor Labs. Austin?
Yes, I'm Austin Terwilliger, the Director of Operations for Taylor Service Center here in Houston, Texas. I manage everything that is needed to get phage cocktails to patients.
And then Justin.
Hi, everyone. I'm Justin. I am the lead data scientist for Taylor. Basically, my job is to look for bad things in phages to make sure they're clean so we can put them in patients, as well as anything else that involves a lot of numbers and a lot of data.
And I brought Keiko here, so she is also a member of our lab, and she works on phage and bacteria coevolution too.
Nice to meet you, Keiko.
Hi, nice to meet you too. Thanks for having me join you guys.
And today we have, as a guest, who should come often, Josh Borin, who recently published a paper. Josh, do you want to introduce yourself?
Sure. Thank you guys so much for having me. I'm happy to join. So just finished my third year as a PhD student in Justin Meyer's lab at UCSD. And I also have been kind of collaborating with David Pride's group, who runs a bulk of the research arm for IPATH, the UCSD Phage Therapy Center.
And you just published a paper. That's super cool.
I just published a paper, yeah, it was nice. Thank you.
In PNAS on phage and bacteria coevolution and phage training. So, do you want to introduce us to the concept of phage training and why phage training is important for phage therapy?
Yeah, I'd be happy to. Ever since I kind of, became interested in molecular and microbiology towards the end of my undergrad, and that was kind of through the portal of experimental evolution, bacteriophage coevolution, the reciprocal adaptation of kind of host and phage, as well as the idea that you could kind of like use evolution as a tool to make your phage better at killing the bacteria, was always really appealing. The motivation, behind that is, I guess, kind of poignant in phage therapy because bacteria evolve resistance to phages so quickly. It seems like it's as quickly or even faster than antibiotics. And so I think that, there's a broad front of efforts and ideas on how to improve phage therapy and kind of like getting your phage to better kill the bacteria and or kind of delay or suppress the evolution of resistance was kind of our motivation behind trying to train some phage.
And how do you train phages?
So, I think our approach is just one of kind of many that has been attempted. The Appelman's protocol and kind of like, so essentially, I think phage training is a broad umbrella that, encapsulates any way that you're kind of having a phage adapt to better infect some hosts. So there have been kind of the Burroughs Appelman's paper where they're using recombination between phages as well as adaptation to kind of get those phages better at infecting hosts, but also infecting new hosts. There have been attempts at, I guess, what I kind of call evolving against a non-evolving host, where you would have the phage grow with the bacterium each day, and then you kind of extract the phage from that culture and replenish the host, the bacteria, from a frozen stock. So in that sort of way, the phage is always continuing to experience the kind of same host, the kind of naive host, you might picture it. So in ours, with coevolutionary training, in some ways I think it's the simplest way. You kind of leave it up to the phage and the bacteria. You put them both in a flask culture. You let them grow together, and then each day you transfer a portion of that flask into new media, and you kind of just let them propagate together over time.
And then you sequence all of that?
I'm sorry?
And you sequence all of that and do EOP and OPS assays and look at PFUs and...
Yeah, so the first thing in this project, and it was actually kind of how to win over my advisor, Justin Meyer. He, I guess, was a little bit reticent to undertake a phage therapy project, or project motivated by ideas in phage therapy, and part of that is because we often see resistance evolve against our phage. So the entire lab so far works on lambda phage. We work on kind of model systems, but typically the bacteria get resistance to all the kinds of lambda phage that we've used in like very, very quickly, in a matter of hours to days. But I had started working with this strain in the lab, this kind of trained phage that I noticed would leave cultures clear for days on end with this bacterium. And so it wasn't until I collected probably a week's worth of data, so kind of like from figure 1, where we're looking at the density of the bacteria in these flasks with the phages over time. And what I kind of wish I could have put in the paper, I don't know, maybe in the supplement or something, is that when you look at those flasks that are at like 10 to the 4, maybe 10 to the 5 cells per mil, that they're essentially clear. And so it was really awesome to have this experiment where kind of control flasks with the untrained phage are totally turbid with cells, and then you have these flasks that day after day remain clear cells.
A cool picture, but in this paper, there should be pictures. So, I thought that, okay, how did you train the initial lambda phage that you used?
Sure. So fortunately, the trained phage in the manuscript was actually trained technically back in 2012 in my advisor Justin Meyer's PhD work. So he ran a coevolution experiment where he took this lytic form of lambda and E. coli strain, E. coli B strain REL606, and he put them in a flask together. And this is a minimal glucose medium. And then every day he transferred 100 microliters from this 10 ml flask into a new 10 ml flask. And that experiment went, I think, for like 37 days. And so over the course of this coevolution, so the bacteria is evolving to resist the phage, and the phage is evolving to better infect its host. Typically on around between days like 8 and 15, lambda phage evolves to use a new receptor. So kind of natively, lambda uses a receptor called LAMB. That's a maltose porin on the E. coli cell. And the bacteria rapidly evolves resistance in getting rid of LAMB. And kind of preceding that, the phage evolves a number of mutations that eventually allow it to use this new receptor called AMPF. And AMPF is primarily a glucose porin for the E. coli cell. So I kind of wish I could draw kind of a timeline. So you have the phage on a lineage that's using this old receptor LAMB, what we think happens is then as the hosts gain resistance, the phage is kind of pushed to use this new receptor on Beth. And then what seems like happens is the phage gets much better. It kind of gets acquainted with its new ability and gets much better at using the new receptor in conjunction with its native receptor. So it can use two receptors.
So phages were able to use as the receptor, LAMB as a receptor, can now use the Beth, but phages that use Beth, are phages that use Beth as a receptor able to use LAMB?
Are phages that use Beth able to use the original receptor, you mean?
Yes. So is it the idea of having two possible receptors, or is it just a change in the affinity of the tail?
Yeah, so it's actually that, so our trained phage in the paper can use both receptors. So we call it kind of a generalist phage. We have a kind of diversity of different generalist phages that use these two receptors, but they all kind of behave differently. They all have different, what we call specializations on one or the other or both, meaning that kind of through adaptation, different experience, these generalist phages might evolve to prefer one receptor over the other, but all of these kind of generalist phages can use both receptors.
That's cool. And did you, I don't know if I remember correctly, but were you able to look at the efficiency of plating of the original phage versus the new evolved phage on the strains, on the original and the evolved strains?
So the question. So we kind of, I guess the perspectives, there's kind of like three kind of time point perspectives to look at the experiment. You have the untrained phage, then you have a trained phage that we kind of start our coevolution experiment from, and then that goes forward from there another 30 days. So we found that the EOP, if you take our trained phage and you look at the bacteria that evolved kinds of resistance against the trained phage was able to evolve kind of higher EOP and get better at resistant hosts. We never measured kind of the EOP of the trained phage and the untrained on the kind of original target host, this E. coli strain REL606. I can say that our untrained phage, the kind of more traditional lambda phage, grows to higher titers on this host than our trained phage. And we think that the reasons for that are twofold. One, the trained phage has a much, much higher growth rate and kind of like, you could call it higher fitness on this host. And so it tends to kind of burn through and kill the host really quickly. The other reason we think it reaches like lower, like if you grow an overnight culture, lambda, traditional lambda, untrained lambda will be like 10 to the 9, maybe 10 to the 10 PFU per mil, and our trained phage somewhere like 10 to the 6, maybe 10 to the 7. And so another reason for that, that another PhD student in the lab's research is focused on really heavily, is that when these phages evolve to use the new receptors, so they're now generalist phages that can use two receptors, they start to become unstable in that they kind of decay at a higher rate. So we kind of think of, before joining this lab, I always thought of phages as like you have these fridge stocks, right, not even frozen, that can remain viable in the same concentration for, years to decades. But we find that some of these generalist phages actually begin to degrade more like on the order of weeks to months. And so there's been kind of a lot of work in the lab trying to figure out why that is, but it seems like acquiring the new kind of function destabilizes the protein a little bit. Sorry, that was a
that is super interesting.
long-winded answer to the EOP question.
No, that's super interesting because it also implies that at the end of the day, phages that are able to bind to more than one receptor have probably tail fibers or proteins that are also not being able to maintain their properties in the population. And that also means that these generalist phages, or could mean that generalist phages that bind to more than one receptor are not common in nature.
Right. Yeah. So there's kind of there's a partnership, a teaching faculty that was a former postdoc in the lab, in Justin Meyer's lab, just kind of partnered with C phages, this like widespread teaching module to look as they kind of isolate natural phages from the environment, try and see if certain phages are unstable and if that correlates with broader host range. The idea being that maybe we think that there's this trade-off kind of between some phages' host range and maybe their stability. And so that might be, why a lot of the phages that we find use single receptors.
And also, maybe why the idea of having phages that infect multiple hosts or are more generalist, their fitness is lower than the phages that are able to infect the bacteria using one specific receptor.
Right. Well, I do, think that fitness is kind of how you define it. And so, like, in nature, this is all hypothetical, but you could imagine, an environment where a phage encounters a host for short periods, and it's really productive, and then it has to kind of sit and wait around. And so during that kind of hiatus, you would really want this phage, or the phage would want itself to be like stable until it can find its next host. What we end up doing in the lab through these experiments is essentially the phage has, quite an abundance of hosts and pretty consistently. And so we think of that kind of trade-off easing up, where this phage has a reliable stream of hosts and kind of propagating itself. It doesn't have these long periods where it might go without. So its fitness in these lab experiments is much higher in that the phage can kind of propagate much faster than if you put both of these phages in a flask together, you put the untrained traditional lambda phage and this trained phage, the trained phage will outcompete the kind of traditional one because it will get to hosts and use hosts and burn through them much faster. But presumably in the wild, the traditional lambda phage maybe is a bit more stable, say in your gut or something, can wait around for its next host. So
makes sense. So, Justin, you had a question. Justin Mother, you need to unmute yourself.
Can you hear me?
Yep.
Great. Okay, awesome, thanks. So if you have a LAMB receptor and then a glucose receptor, have you tried to make a knockout of both of them and see how generalist it really is?
Yes, awesome question. So the strain that we typically use for these coevolution experiments, REL606, does not have OMP-C, which, for example, like K12 E. coli does. So OMP-C acts as an alternate glucose porin. And so the issue with REL606, the bacteria we've been using, has been if you knock out both LAMB and OMP-F and you culture it in a minimal glucose media, the cell is like really sick and it can't get carbon. And so the experiment doesn't really work. People have kind of engineered that strain to engineer OMP-C in and remove OMP-F. No coevolution experiments have been done on it yet. We do know that, we haven't found these kind of generalist phages to use a new receptor yet, and people have kind of looked around a little bit for that, but that is very much kind of an outstanding question in the lab.
What about like LAMB homologs like in Shigella or other Enterobacter?
Also, great question. I wish we had done more work on. I have given the train phage to an infectious disease fellow in IPATH, and I believe she found that it could infect certain strains of Salmonella, but I would have to talk with her about details.
Thank you.
Thank you.
Archana, welcome. You have questions?
Yeah. Hi, Josh. And I have a question about the transient phage resistant mutant. So, what's your opinion about transient phage resistant mutant? And I just want to know, like, if they do carry any mutated gene.
Yeah. Could I ask? Are you talking about how there was like a bit of resistance?
The phage resistant mutant that revert backs after serial passage.
Yeah. So, kind of looking at Figure 2, Panel D, if you had that open in front of you. Maybe I'll give a little background, and please interrupt or correct me if I'm not answering the question. So, over kind of 30 days of coevolution of the train phage with the host, we later went back to kind of freezer stocks of populations from each day, and then we isolated bacteria across time and looked at when resistance evolved. And specifically, we took these bacteria from days 5, 10, 15, 20, and tested them kind of against the trained phage or the respective phage from the treatment. And we ended up doing that in like liquid plate reader assays because we found that the EOP wasn't quite as sensitive as like looking at changes in the actual growth curve of the bacteria. And in doing that, we found some bacteria were, kind of traditionally completely resistant in that there was no difference between how the bacteria grew, whether the phage was there or not. We found a lot of bacteria that had what we call partial resistance, in that the bacteria would actually have some visible growth, often kind of like a hump in growth during the plate reader assay, but nowhere near kind of what the bacteria would grow like without phage. And then we also found a lot of persistent sensitivity where we just didn't see any growth. That doesn't necessarily mean that the bacteria weren't growing better than the kind of ancestral sensitive. It just means that it was kind of below the sensitivity of these plate reader assays. So I think what the question was, and kind of what was, a very interesting and confusing part of this test was, in one of the 3 populations, on day 25, there was all of the kind of 12 isolates, bacterial isolates, were partially resistant. And then on day 30, they had all kind of, I guess I wouldn't say they reverted to sensitivity. I would say the 12 isolates we grabbed were sensitive, meaning what could have happened is that you had a sensitive bacterium on day 25 that was at really low frequency that we didn't detect, and this partially resistant one that was at very high frequency. And on day 30, those had kind of switched, where you now have the sensitive type that's dominating in the flask. So I have there's 2 working hypotheses, I guess, for why this might have happened. That all of a sudden, how is sensitivity dominating? How are the bacteria growing when, there's still phage in the flask and they don't appear sensitive? One of them is that the phage, we found other signatures that phage during the coevolution experiment kind of evolved to infect the resistant host. So, for example, in a different population on day 25, there was a completely resistant isolate, and you would think that would just take over, but it disappeared after that time point in the experiment. And so what I did is took a phage from that same day, and I ended up finding that it had kind of evolved to infect this completely resistant type. So it could be that the phage kind of countered the partial resistance really well, and that kind of eliminated it from the population, and maybe the phage that could infect these partially resistant types, don't do as well against these sensitive types, sensitive to the original trained phage. Sorry, this is turning into such a long-winded thing.
Actually, it's so interesting because I did find the same result to my Staph aureus. I'm working on Staphylococcus phages. So what I did, I isolated my phage-resistant mutant in January. But now, after lockdown, storing the phages in the freezer, now I'm finding the phage has turned sensitive. So I just want to know, like, if I sequence them for whole genome, will I be able to find any resistant mutant genes, or will it be same like wild strains?
In the bacteria?
Yeah.
Is what you're asking. That's a good question. And so the bacteria have been in the freezer, not in the fridge?
No, it is in -10.
Okay. I guess, so there's a lot of people in the IPATH lab right now that are running experiments where they challenge a bacteria with phage. They see resistance evolve, and then they're sequencing the bacteria whole genomes, trying to look for what the kind of resistance mechanism or mutation is. I think a lot of them are having trouble with, if you don't have a really good reference genome that's annotated, it can be hard to identify exactly what the kind of the focal mutation is. They're also finding, and I found this too, that when you sequence these genomes, sometimes the bacteria get resistance by deleting like multiple genes, or even like thousands and thousands of bases, and so that can kind of cloud your ability to identify what it was. As far as the as far as the sensitivity resistance status changing, Dr. Phriday, who runs the IPATH lab, had actually alluded to that in lab meeting yesterday, that some people were finding resistance status changing over time, and he and people in the lab weren't sure why that was, but I'm pretty unfamiliar with that.
And what's your opinion about the difference in the DOP between the wild strain and the injection?
Could you elaborate a little bit?
Yeah, like after isolating the phage-resistant mutant, I found that the mutants had different EOP when compared to the wild strain. So, what is your opinion on that? What could be the real reason?
So you have a phage that infects a bacterium, and then you have an evolved phage that infects the bacterium, and the evolved phage has a different EOP on on which bacteria? On resistant bacteria?
Yeah, on resistant bacteria, it had a different
EOP. I guess if I understand the experiment, my thoughts would be that the phage is evolving to better infect the resistant bacterium, and so its EOP might be changing compared to the ancestral phage. So we.
Thank you.
Thank you.
Anybody else have more questions? Because I can talk a lot.
I have a question. So it took 18 days for Dr. Meyer to make that mutant, and your experiments are 30 days long. Why so long?
Yeah, that's a great question. I think that, so it takes a while for the phage to get use of this new receptor. We found that dual receptor use was kind of these generalist phages, that effect was really driving this kind of prolonged suppression and resistance. So I think that you can pretty reliably say that in our system, being able to use two receptors was really important. That takes quite a bit of time because And so there's been a lot of work digging into the kind of coevolutionary dynamics that lead to the new receptor. A recent PhD student, now postdoc in the lab, has submitted a manuscript. I think it's on the archive, like really digging into that. But what it seems like happens is that the phage initially isn't that good at suppressing the bacterium. And so it takes the phage like 2 mutations to get much better at infecting the bacterium. And when it gets to that point, the bacterium now has a much larger incentive for evolving resistance. And once it does that, then the phage starts adapting to use the new receptor. So in terms of like why it takes so long, in large part is just kind of waiting for all of these dynamics to play out. And then I think the second part of that is, we know that the second the phage evolves to use 2 receptors, it's still not that great of a phage. I think of it as kind of like it's evolved this new function, but it doesn't really know how to use it that well. And so then it, takes a number of days, I think, for the phage to adapt and become much better at using this kind of getting used to being able to use both receptors on these hosts. There is a project getting started in the lab by a master's student that is interested in, I think what your question was getting at is like, can you speed this up? Like, patients can't really wait for you to train something for 30 days.
What it is. Yeah, because, we deal with patients, and so we're, wondering how fast can we train a phage so that we could see this effect.
Yeah, so their idea is kind of a pseudo bioinformatic approach of like, how can you get a lot of information about the kind of training quickly and then kind of like speed that up through engineering. So kind of a combination of the two. I have always been really interested in the idea of, I think it makes sense given the kind of effort and time it takes already to get approval and find phages that work and clean them up and make clinical preps and deliver them to the patient is like already too much time, and people are trying to speed that up. I've always been really interested in what would happen if you train the phage kind of in parallel. So you start treating the patient and you're training the phage on the side and then give that to the patient, when it's available. Obviously, maybe that should be trialed in like a mouse experiment or some animal model first, but I've really been interested in seeing if that works.
I mean, we are purifying the phages in the actual patient sample, and so the patient's bacteria, but, it's only for like a few cycles, right? It's not.
I see.
It's not that many days. But I wonder if that also is kind of a good thing to continue purifying on the patient bacteria, but also using this technique of training phages too.
Yeah, I would be. Have you tried sequencing like the original phage and then after those like maybe 3-4 cycles on the patient's bacteria?
That's something that we're thinking about doing. But no, not yet. So
okay.
we usually just sequence after purification.
Okay. That would be really cool, especially, if you have all these different, phages and species of bacteria, different strains, and can show that, I would imagine it's a general phenomenon that the phage are at least getting, a few mutations, because they evolve just so fast. And if it's a new host, I feel like there's always some ground to be gained. That would be awesome.
There's also a lot of factors into account when you're thinking of this in the context of phage therapy, and it's not just the fact that you're putting phages in contact with bacteria, but you're also having to take into account the host immune system and the pressures that the host immune system has on the population of phage that you actually use, and how many of those phages are actually reaching the target bacterium, and the phages that result as the infection of the hosts progress, and the new bacterial strains, which is super cool. I I think this is something that should be looked at in depth. Varun, you had a question?
Yeah, hello everyone. So my question is to Josh. So as you mentioned that the new trained phage is using a different receptor than the previous one. So I had a question like, did you find just only a single change in the receptor, or there were many other receptors used by these new trained phages? Like, did you find other cases as well in terms of like this aspect?
So if I interpreted correctly, you're asking whether the trained phage can use just 2 receptors or whether it can use maybe even more than that?
Yeah, like how many different type of phages were evolved after this training, right?
So, we've only found the kind of these two receptors being able to be evolved to use. However, we do find that it is really repeatable. So, in a previous study, they did, I think, 96 like microwell populations, and they ran this kind of coevolution between the original lambda phage and the host. And I think they found that it evolved to use this new receptor. So it evolved to use 2 receptors in, I think, like 28 of those populations. So it's quite repeatable, but we've never found the phage evolved to use a different second receptor or a like third receptor yet, which is actually kind of surprising because OMP-F and OMP-C are, I mean, if you look at the protein structures, they look very similar, but for whatever reason, we haven't seen it evolve to use that yet. I would, I guess, kind of echo one last thing, which was that we ended up, in the bulk of the study that we published, we really focused on kind of like this original trained phage. That was just the genotype we started out with, and we really kind of focused on trying to figure out why it was better and how the bacteria was evolving resistance and stuff. At the very end of the paper, kind of at the encouragement of reviewers, questioned the repeatability of like, did we just pick, is our trained phage just like a lucky pick? And so we grabbed a bunch of other phages that were trained the same way, essentially independent replicate populations. And we found that it didn't seem to matter which trained phage we used if the trained phage had evolved to use 2 receptors. If the trained phage, so you can imagine, you can train a phage and it doesn't evolve to use a new receptor. So we had a panel of those phages, and they were better than, the ancestral phage, but not nearly the kind of effect size that, we got really excited about with phages that could use 2 receptors. Does that answer your question?
Yeah. So, can we prepare a sort of database or something like that will, like, if say resistance while using these phages for the treatment of patient, and if we see some kind of phage resistance against these phages, so we can go and check very initially directly for this kind of mutations which have occurred in the host bacterium. Like, can we do something like that?
Do you mean like predicting which mutations might allow a phage to use a second receptor?
Yeah. Yes.
Totally out of my realm of expertise. I feel like, and other people I think could speak to this, that is, I don't know, kind of like the holy grail that a lot of people really want to understand is like bioinformatically predicting phage host range. I think there was a talk given by Felix Biotech, where my impression is that really that's what they're going after is a lot of bioinformatic techniques, but it sounds like it's a pretty tough battle. I do think that We don't understand in the Meyer lab how often, you evolve to use a new receptor through coevolution. People have really been hammering away at details of this model system with lambda, but if you use other phages, how likely is that, and under what conditions, environmentally and ecologically. But there was a really, I thought, great paper by Courtwright in Paul Turner's lab, and published in PNAS in 2020, I believe, that is looking at kind of transposon methods of figuring out what a phage's receptors are, and I think that it would be a great start to start looking at, can we identify natural phages that use two receptors bioinformatically, and if and when you can kind of find dual receptor phages, then start kind of looking at how their, properties, therapeutic or killing or deferring resistance properties compare with single receptor phages.
Yeah, thank you.
Mm-hmm. Justin, you had another question.
Yes. I was going to comment on maybe a really easy or really tough question. So lambda and lambda B are literally the textbook, receptor phage interaction, right? And can you hear me?
Yeah, I can hear you.
Yeah. So my question is, not all phage and receptor interactions are created equal, and some are going to find plasticity or the ability to evolve to other receptors very easily, and others it's going to be a lot more difficult when the interaction is a lot more complicated. I was just wondering if you could comment, when is this going to be easier to find, and when is it going to be harder to find, for a specific phage?
Mm-hmm. That's a good question, and I think that lies more on the tough side of the fence than the easy side. So I think that my impression, and I haven't worked with many LPS binding phages, is that some phages appear more plastic in their host range. My impression is that those are ones that use like LPS and these kind of complex long chain molecules as phages that use.
Well, you got to be really careful too, because some of those interactions are enzymatic. Some of them are actually chewing up antigen and might have a lot of things that could potentially need to be maintained or could be potentially screwed up by a mutation.
Right.
So I'm just trying to think of a way to model this in my head of like, okay, if there's only a few binding sites, we think about it in terms of binding sites or time evolved in the receptor.
I see.
What's the right way to think about this?
So our kind of working theory in the lab is less about, we're trying to get a crystal structure of lambda's tail fiber tip protein J, and I think that would give us a lot more power in testing hypotheses. But kind of our kind of heuristic or empirical hypothesis is that so the phage gets these mutations on the way to evolving to use the new receptor, and what those mutations appear to do is two things. One is that they make the phage's tail fiber protein less stable, and so we kind of wonder if this is conferring some sort of flexibility in the tail fiber that is allowing it to maybe conform to different shapes in the receptor or allow it to kind of exploit binding sites that are maybe a little bit more, in different positions. The other thing that we've noticed and are actively looking into is an idea of non-specific binding, that when you get mutations in a protein that was previously highly specific, it seems like kind of pleiotropically what you get with instability is also a protein that starts to bind things less specifically. So it seems like some of these phages, before they get the ability to use the second receptor, they start binding things on the outside of cells that aren't just LAMB and aren't OMPF. So we've taken these phages kind of en route to using the new receptor, and we've looked at how well they absorb to double receptor knockout hosts, and we found that they absorb better, appreciably better than kind of the ancestral phage. So I think that our expectation is that the phage's protein is becoming kind of more flexible in both, I think, a biophysical and evolutionary sense at exploring kind of new receptors. And so I guess how I would extend that is protocols or ways in which you can get a phage to become a bit more promiscuous in its kind of host recognition protein might allow it to broaden its host range or use new receptors. And I would only be comfortable, I think, extending that to like these traditional lock and key protein-protein interactions. Enzymatic ones, I have no idea how that would work.
Well, this is very fun, a very fun line of thought. But I think as I think about it, I'm like, wait, is there a way to make a phage tail spike or tail fiber more stable, so it'd be more agreeable to evolution?
Right.
If you could strengthen it, then it would be, more adaptable later on. Anyways,
this is great.
Great stuff.
Yeah. So I want to just briefly highlight that, I think appears to be so. There's a PhD student, fourth year, and she is actively writing up and submitting a paper that is actually directly testing this hypothesis. So I think the prevailing one that has kind of been championed by Francis Arnold is, this idea that if you engineer in stabilizing mutations, the protein then is able to kind of become destabilized by neofunctionalization proteins to kind of allow the proteins to adopt new functions. I think it's also important to keep in mind that most of the proteins under this kind of hypothesis are catalytic proteins. So the idea being that you stabilize a protein not in the active site, and then the active site can kind of acquire destabilizing proteins and do new functions. So the working idea in our lab goes kind of counter to the established Nobel Prize winning hypothesis, in that at least with these kind of structural and like lock and key proteins, that destabilizing proteins might actually be the ones that are conferring new function. So this grad student in the lab has found that when you insert stabilizing proteins into the tail fiber J gene, or mutations into the tail fiber J gene, it actually reduces or completely eliminates the phage's ability to evolve to use the new receptor.
My brain's going crazy with all of these different ideas, but you guys have lots of other things to talk about, so I'll be quiet. But thank you very much.
Thank you very much.
I actually posit that this the changes and the frequency of changes and the stability and the ability of the proteins to acquire new functions, or at least bind to new receptors, would change if actually the protein is interacting, the tail fiber is interacting with a protein on the surface of the bacteria versus a polysaccharide or anything that is a polymer complex that actually can change the number of units available, like the O antigen, or like exopolysaccharides, any type of exopolysaccharides where the proteins would bind and then have that catalytic activity that Justin was talking about, because they require the catalytic activity to actually affect.
Right. Yeah, exactly. I think that's exactly.
Well, does anybody else have any more questions?
I have one question. So I'm thinking about what you're saying with stability and sort of the reluctance, so to speak, of Lambda to sort of jump to more than the receptors that you've already found in LambB and also in OFF. And what I'm kind of wondering is, because phage lambda is so, what's the right word? It's a laboratory phage, right? It's been around for a really long time and replicated for a really long time, basically all the same way. Maybe it's lost some of the plasticity that it might have had in the past. And I was wondering if you thought that maybe using phages from environmental samples that are more close to a wild-type phage would be more plastic and more amenable to evolution.
This is all such great questions. I'm so happy to be talking with people in this group, because that question, it kind of can be tested with Lambda. So let me back up. So Lambda when it was originally isolated, actually had side tail fiber proteins. If you already know this, and I'm just kind of like, I don't know, reinventing the wheel, please stop me. But so those side tail fiber proteins appear to function in binding to OMP-C. And this phage lambda, as most labs have it, or most labs that have lambda these days, has lost those side tail fiber proteins. The reason being, and there's a paper published, I'm forgetting the name.
Lambda Papa and Lambda
Right, exactly. Your lambda and lambda papa, which lambda papa is the one without side tail fibers. Your lambda had them, and the idea is that it made larger plaques once it lost these side tail fiber proteins for whatever reason, and so that one was kind of picked and propagated and disseminated into the research community. So I think that as kind of like just thinking about the lambda's true history as, supposed wild-type phage in the human gut, being that it had a primary receptor through which it could infect LAMB, but it also had side tail fiber proteins that could bind OMP-C, that could bring it in and kind of help it absorb to cells. So and this is, well understood and widespread, that phages can use a co-receptor to kind of like locate it to the cell and absorb better. So we have wondered what would happen if you gave lambda back those side tail fiber proteins, if that would kind of like help or hinder this host range expansion and stuff like that. But I guess getting back to your main question is like, how would the ability to evolve new host range differ for like a lab versus wild type strain? I don't really have a great intuition. I've inherited a bunch of lambdoid phages that I believe vary, and kind of none of them are the kind of like as big of a lab rat strain as lambda. But I don't know how much they vary and how long they've been propagated in the lab. But I am going to be trying to kind of see through a series of projects led by undergrads or whether they can evolve to using a receptor. We'll see.
Cool. That makes, that kind of makes sense to me. Do you think then, I would kind of guess that phages with a secondary receptor would have a better chance of more readily adapting to a broader range of targets because they have more than one way in which they can interact with the target and adapt to that. What do you think about that?
Yeah, so in this, before with Justin, we were talking about this like non-specific binding idea. And there are a number of experiments in the lab that indicated but weren't concrete and repeatable enough for us to like publish it yet. And so it's still kind of being sussed out. But we expect that in a lab culture, so let's say you have E. coli and phage lambda, and lambda can bind, say it can bind to two receptors, but it can only infect through one. But every cell in that flask is a potential real host for phage lambda. So there's kind of a selection for, or like a benefit of lambda to having kind of this promiscuous binding to whatever really on the outside of a cell. Because once it kind of lands on a cell and takes hold, it can then kind of like, search around that cell for its receptor that it actually infects through. We have this, general unsupported theory in the lab that in more natural populations, you can imagine phage lambda trying to find E. coli in the human gut. If it binds non-specifically to, say, a bunch of different receptors, what that might end up meaning is that the phage spends a lot of its time bound to molecules on the outside of cells that aren't even, potential hosts. So there could be this cost of being too promiscuous in a true ecological scenario that doesn't exist in a lab culture when you just have one host type. There was a paper published by Israeli group in Scientific Reports, I think in 2019, with T. either T4 or T7, where they looked at, how does a kind of diversity of hosts that the phage cannot infect, impact the phage's host range, and they found evidence to support that. You had a kind of contraction of the phage's host range, and you had a contraction of what the phage would bind to, because it was, I think, binding to things that it couldn't infect, was my interpretation of their results.
That would make sense to me. Just generally lowering the encounter rate with a potential target, actual potential target.
Right. But with the kind of evolution of using the new receptor, there's, I guess I'm probably stating, the obvious, but you have to have, the phage has to be able to interact with a new receptor in order to have, the kind of variation even to be selected for. So there is this, I think ecology is playing a pretty paramount role that I definitely don't understand really.
Well, I would think that in the wild, sure, what you were saying about binding too promiscuously to potential targets that they can't actually infect would be detrimental. But in the lab, we do have the luxury of putting together exactly what we want together. And so I would think that having those extra tail fibers to sort of broaden the number of cells the phage can interact with in one way or another would be to our benefit.
Right. Yeah. We definitely should use your lambda with OMC, and then maybe that's all it needs to now we know it interacts with OMC, and maybe it can evolve to use it.
Yeah, I'd be interested to see what would come out of that. Anyway, thank you.
Thank you.
John, nice to meet you. Do you have a question?
Thank you, Adriana. Actually, no, I do not have a question. I've been listening to this group with so much pleasure. I envy your expertise on this, and if somebody would ask me, although I'm 69 years old, what would you like to be when you grow up? I'd say, like any of these people in this group, thank you so much for all this.
Thank you for joining us. Thank you for joining us every Friday. And you are more than welcome to join us next Friday when we're going to continue this discussion.
Yes.
I hope Josh can join us as well.
Yes.
We have another evolutionary biologist coming over and talking about phage and more phage evolution.
Yes.
So it would be
awesome.
awesome.
I'm also hoping we're going to be joined by people from the Eliava Institute, because Phage Training has been around for a long time, 80 years. So I'm hoping they can also provide some expertise. And we had one more question, I think.
For a while, so their expertise is highly valued.
I know. I'm worried they're going to say, we already know all of this. This is old news.
Well,
this is such an awesome episode. Just want to chime in. We're going to publish Josh's behind the paper. Hey, Josh, by the way.
Hey.
Thank you. So, yay. This is you can stay tuned for more. I'm so excited to help promote it. Such cool work.
Thank you. Thanks, everyone, for coming and bringing such great questions. This is awesome to talk about.
I think we have time, a couple of minutes for one last question, which is Tao.
Thank you for having me in the meeting today. So I just have some questions. Maybe, do you think there are phage species that we cannot change? Because now I read the paper about phage training, and they already public about the E. coli or Pseudomonas aeruginosa or something, but there is no paper on Klebsiella species or Acinetobacter species. So do you think is there some phage species that couldn't be changed?
I guess I like to think that kind of a broad enough diversity of phages can it kind of infect each species of bacteria. So I would think that it's not necessarily about the species of bacteria can the phage be trained to, but rather, which phage. For example, as we were kind of highlighting earlier, like if you use an E. coli phage that infects with LPS, its ability, those phages that use LPS as a receptor might behave quite differently than phages that use a protein porin. But I think that the kind of bias in papers on E. coli, I mean, in our case, we're very much on the kind of lab rat side. We use model systems so far, and people using Pseudomonas aeruginosa, really highlighting areas of phage therapy that would benefit really heavily from training or, better phage therapy, like, patients with cystic fibrosis and these long-hauling antibiotic-resistant infections. But I think, you should do one on Klebsiella.
I can also kind of chime in on that and tell you that most of the strains that are clinically relevant of Acinetobacter and of Klebsiella are encapsulated strains, so produce exopolysaccharides, and those exopolysaccharides, so although, for example, in the case of Acinetobacter, most of the clinical relevant strains are clonal, so they are part of one of three clones. The highest point of diversity, or one of the highest points of diversity for these specific clonal strains that have been reported in outbreaks are usually the O antigen locus and the K locus, which is the O antigen encodes, the O antigen locus encodes for the polysaccharide that's actually the O antigen, and the K locus, it's the locus that encodes for genes that code for the capsule locus, for the capsule of the bacteria. In the case of Klebsiella, there's also a large variation in terms of the exopolysaccharides that they have outside. So unlike the case of Lambda, where you have a protein interacting with a protein on the bacterial membrane or a porin in the bacterial membrane, in the case of Acinetobacter and Klebsiella, the more likely scenario is that phages are going to have to be interacting with these polysaccharides that are polymers, that are more complex, and they're also, because they're not necessary, they're not essential genes, the bacteria tends to be able to mutate them faster. And even small changes in these exopolysaccharide genes can actually lead to larger changes in the polymer per se, hence the resistance. So I think that training phages in those cases is different. I personally use the Appelman's method to try to train phages against Acinetobacter clinical strains, specifically the ones that were obtained from San Diego in the Patterson case. And it was not as easy and evident. It didn't work as easily as it would for Salmonella or E. coli.
Yeah, thank you for your answer. And I have one more question. Did you check the bacterial genome before using it for training the bacterial phage? Because I think sometimes we can find the prophagine in the bacteria. So in case it was induced and become a free phage, so have you ever experienced with that case?
Yeah, we know that there's no functioning or fully functional prophages in the bacterial genome. There are prophage genes, which we had some interesting interactions with. But if you induce the strain of bacteria we use, no phage pop out.
Yeah, thank you. And just the last questions. Can I ask about have you ever met some case the evil phage, the chain phage that you can isolate it? Have the phenomenon of lysis from without, like it can kill the bacteria, but there is no production of phage.
Yeah, I think for this phage, I don't think that it can do that. I'm actually not that. My impression of lysis from without was if the phage has some sorts of endolysins that it's producing. Maybe someone else.
Not necessarily. If you have a large amount of phage, that actually can hinder bacterial growth or cause stabilization of bacterial membranes just for unspecific binding, and that would cause lysis from without, not necessarily referring to endolysins or anything related to that. It's just a matter of artificial MOIs on a fluid membrane.
Like if you have phage that essentially plug up all the receptor or bind up so much LPS that the cell can't function.
Or phage on the culture, and it's not binding specifically to anything, but there's just so much that it ends up sticking to surfaces, and that destabilizes the membrane.
Okay. In our case, I think the MOIs would not be quite high enough to do that, but it's not something we look for. So.
Yeah, thank you so much for your answer. Thank you for having me here.
Thank you.
Yeah.
Jean-Paul, you were going to say something?
I just wanted to add that I think lysis from without can also be obtained when the phage binds, injects its DNA, but then it's not able to hijack the machinery of the bacterium, or it's broken down by CRISPR-Cas, for instance. But punching a lot of holes then makes the cytoplasm come out of the bacterium and killing it.
Okay, so I think we're 9 minutes past the hour. Josh, thank you so much for staying the extra time. Thank you, everybody else, for staying here. We hope you can join us next Friday as well. It's going to be at the same time. So 11 a.m. for you. I'm assuming you're in California. 11 a.m. for you, and for everybody else, for Central Time, it's going to be at 1 p.m. And for Eastern Time, 2 p.m. I hope to see you all next week, and enjoy your weekend. Have a great Phage Friday.
Bye, everyone.
Thank you. Bye-bye.
Great talk. All right, bye.